So Cheyenne from Cornell is actually going to be telling us how you can extract strawberry DNA on your own at home. So just as we were talking about the RNA and the DNA in the cells, now you have a chance to do it yourself. So I'll hand it off to Cheyenne, who's ready to take us through a really cool activity.
Thank you, Barrett. So as we're doing the demo, if you have any cool pictures of your DNA that you extract from your strawberries, go ahead and send a picture or post with these hashtags here. Oh, excuse me. These hashtags here. And I'll leave this up to the side here, and we can start extracting our own strawberry DNA. If you don't have strawberries, you can use other fruits, but strawberries will give you the most DNA because they have 8 copies of all of their chromosomes, and they just produce a lot of DNA. So first, you want to have, you can do this with as little as 1 strawberry. I have 4 in here. Put them in a plastic bag and seal the bag. And what you're going to want to do is you're going to want to squish these. This is probably the most fun part. Just gently squish the strawberries until they're kind of a goopy liquid, until you don't have any big pieces left anymore. So this could take a couple of minutes. Just gotta get all the big chunks out. Make sure to be gentle so you don't explode your bag. Make a mess everywhere. All right. So now your strawberries should look something like this, just kind of a goopy red mess. And then now we're going to make our extraction buffer. So what this is what's going to really like draw the DNA out of our strawberry chunks and liquid. So take a glass. We're going to use a teaspoon for measuring and another spoon for stirring. And what we're going to do is we need a teaspoon of salt. And so what the salt does is as we're trying to extract the DNA from the cell, it helps break it up and it actually remove the DNA from the cell. So we're going to add 1 teaspoon of salt. And then next, we're going to add dish soap. Any kind of dish soap will work, or any soap. And what this does is that cell has like a membrane on the outside, and soap is very similar to that membrane. So it likes to dissolve it and open it up so that we can actually reach the DNA. And so we're going to add a teaspoon of this as well. All right, and our last ingredient here is water, just to mix it all together. I have about a 1/4 cup of water. And we're going to give this a good stir. We don't want to form a lot of bubbles, so stir a little slowly, but make sure everything's really well mixed. And this is our extraction buffer. So again, the soap is to break the cell open, and the salt is really to get that DNA out of there. So this brings us back to our bag of strawberries here. Give it one last squish. And I'm going to add a few spoonfuls of our extraction buffer. So I'm going to add probably 3 or 4 of the spoonfuls. And we're going to reseal our bag. And you want to gently squish again just to mix everything. You want to try to avoid forming a lot of bubbles. So we're going to gently squish our strawberries with our extraction mixture just so it gets all incorporated. Alrighty, so we have our mixture there. And now we need to filter it out so we don't have all those like strawberry chunks in there. We just want the liquid from this that contains our DNA. So what we're going to do to filter it is we have coffee filters. You can also use a strainer. I'm just going to kind of fold this coffee filter up a little bit and place it into my cup. Pour some of our mixture into the filter paper or to our coffee filter. Alrighty, so it should kind of look like this, and it might take a little while to start dripping. So this is probably the longest part of the process. And you want to make sure, you could squeeze it a little bit, but if you squeeze too hard, you might break your coffee filter, which I've done before and got strawberries everywhere. So not the best thing to do, but we see we can have some of the liquid is starting to drip through, and that's perfect. That's what we're looking for. So you can see. So in there is our soap, our salt, and dissolved DNA. And it's dissolved in the water that we added to the mixture. And so the step that we're going to do next is we're going to add isopropyl alcohol, which is rubbing alcohol. 50% is okay to use, 70 or 90% works better, especially if you've cooled it. And what that's going to do is the DNA is soluble in this water here. That's why it gets to drip through the coffee filter. But it's not soluble in the rubbing alcohol. And so what that means is that it'll come out as a goop or as a solid so that we can actually collect it. So we'll take a few more minutes to collect our strawberry liquid. And if, any questions so far can be sent through the Q&A.
So Shane, we do have one question.
Yeah.
The question is from Bailey, and it says, can you use hydrogen peroxide?
You cannot use hydrogen peroxide for these. No, good question. You can use ethanol if you have some of the, instead of rubbing alcohol, some people use ethanol for like cleaning scrapes and whatnot. Ethanol would work.
Cool. And then Sadie is wondering, with the strawberry DNA, what do you do with it afterward?
Good question. So it's just kind of fun to play with. It's really goopy and it's really long strands. And so you can like see it like visibly. But if you, which right now we don't most likely, but if you happen to have access to a microscope, it's really cool to look at underneath a microscope and like really look at the strands and stuff.
Yeah.
Definitely not to the level that we saw in the presentation, but you can see a little bit more detail.
Yeah, another question, I actually had this question myself. So are there any other fruits that you can do this with? Why did you choose strawberry?
Yeah, so other people have done this with like banana, also easy to squish. But strawberries have 8 copies of each of their chromosomes. So because of that, they have a lot of DNA. And then strawberries themselves are a lot of copies of their DNA, and strawberries themselves just have a lot of DNA, so that we just get a lot of it coming up. With bananas or like something of the like, you could do it, but you just wouldn't produce as much of the DNA.
Gotcha.
Yeah.
Okay. And then, oh, this is important because we got to make sure people can do this at home. What is the salt to water ratio again?
Oh yeah, so 1 teaspoon of salt, 1 teaspoon of dish soap, and a 1/4 cup of water.
Cool.
And that's good for about like 2 to 4 strawberries, I'd say.
Awesome. And this is a good question too. Does this damage the DNA when you're extracting it, or is it still intact?
So it's still like kind of partially intact, but the addition of the salt kind of unravels things a bit, and that's why we can pull out these like big long strands and whatnot. So I wouldn't say it's like perfectly undamaged. Yeah.
Cool. Cool. Awesome. And since we did have a couple questions about the ratio of the different supplies, just a reminder that these videos will be posted later. So you guys can always do them again at home or for the first time at home later. Don't worry about rushing to get all the materials correct this first time. Jana is wondering why you don't want to make many bubbles.
Yeah, so if you make a lot of bubbles, it'll just be harder to make. When we pour in our isopropyl alcohol, we don't want the mixture, or we don't want the liquids to mix. We're trying to keep them in separate layers, and so the bubbles will make it a little bit harder to do that. And I'll actually move on to that step here. So I'm just going to take my strawberries in the coffee filter and put it in the cup I had my water in. And we want to be very careful when pouring our isopropyl alcohol so that we have different layers. So that is my strawberry layer here. Here's my cold 91% isopropyl alcohol. I'm going to use my spoon that I used to stir just to kind of press it to the side of the glass. I'm going to pour the isopropanol kind of onto the spoon so it just kind of barely dribbles down onto it. And you want to use about equal volume of isopropanol, if not like 1.5 times volume as you had strawberry liquid. So what this looks like, we have a clear layer on top, that's our isopropanol, and this pink layer on the bottom is our strawberry liquid. And you can kind of see that there's these white flecks starting to come up from this pink layer. That's our strawberry DNA that's insoluble in the clear isopropanol or rubbing alcohol layer. So what I'm going to do is I'm going to help this along a little bit. I'm going to use a chopstick, and I'm going to kind of gently pull some of the DNA up into this layer. And there we go. That is strawberry DNA. We can get a lot more of it out of here
awesome.
too. So there it is.
Very
so, yeah,
cool.
So you can have more fun with it if you have a microscope, but it's also just kind of fun to squish it in your hands. But yeah. Let me see if I can get some more of it going here.
So another question that we had is from Nicole, and she's wondering if it matters whether you use dish soap or hand soap. I assume she means like another gel hand soap.
Yeah. So I think dish soap is the most concentrated, so it's the best for it, but you can definitely use hand soap, and I might just up the amount that you use.
Awesome. Very cool. And just a reminder again for you guys, you can always access this later as well. So you can do this activity at home at any time. And then, I see. So this question is, why can't you mix the alcohol with the strawberry liquid?
So if the strawberry liquid mixes with the alcohol, it'll be, the DNA could be more soluble. Since it is soluble in the water and insoluble in the alcohol, if you mix the two, then it's going to be slightly more soluble and you won't be able to extract out the DNA as well. So it'll be a little bit more of it will stay in the solution instead of coming out.
Awesome. Jacqueline is wondering where you figured out how to do this.
Yeah, so this activity was actually, oh, here's a good chunk of it. This activity was actually done with me when I was in middle school and went to an EYH conference, and that's the first time I did it.
Awesome.
Yeah.
Perfect.
Yeah. Thank you guys for the good questions. And that's all I have in terms of the demo, but yeah.
Make sure you guys share this demo. Cheyenne has those hashtags again up there for you to see, and make sure you share it with everyone. Oh, one last question.
Yeah.
From Saraha, I hope I said that correctly. Would a napkin work for the coffee filter?
It should be able to work, yeah. It might be more likely to break, so you want to be really careful not to like put too much into it or to squeeze it, but that should work for sure.
Awesome. Looks like Barrett is ready.
All right.
You're fine. Awesome.
Yeah, thank you so much.
Thank you, Cheyenne. Thanks for answering all their questions, and hopefully you guys get to do this at home.